I'm Benjamin, computer scientist at Ghent University in Belgium, and we work in a computational lab embedded in a life science institute, the VIB. And the topic of my talk is computer vision on microscopy images. And because I work with computer vision, I always have nice pictures in my talk. So, and then I ask, what's in this image? But this is a bit of a difficult image. It's an electron microscopy image of a human cell. It's nanometer scale. And here, the scientists are interested in mitochondria, the powerhouse of the cell. And the question is, how many mitochondria are here? But it's a bit difficult, this segmentation task. Usually humans are pretty good at segmentation, but here, because it's biology, you need a lot of domain knowledge to get this right. So what do? You ask an expert in the field who studied this, and they'll tell you there were 3 mitochondria in this image located here. And now you can analyze this image quantitatively. You can tell how many mitochondria there are, what are their shape, what is their area. And then you can summarize these statistics to answer certain research questions. And this works for small images, but what do you do when it's a lot larger, when it's a whole cell? These data sets quickly increase in size. You can also see that it's actually in 3D, this data set. And that's a lot harder for the annotators to annotate, and especially because this is not just a single, you don't need a single cell, you want a whole data set where you compare 2 classes against each other. So this is way too much imaging data for a human annotator to annotate themselves. You want some kind of deep learning automated segmentation solution that the annotator can train so that the annotation can be automated across the whole data set. For this, you can use computer vision. There are models out there that work. UNet, for example, is an autoencoder deep learning solution, but it can achieve human level performance. The downside is that you need a lot of ground truth training data to train this on this type of image. And that's a bit of a chicken and egg problem. You have a solution to annotate your whole data set, but for that you need to annotate your data. And you need to annotate it quite a lot, 10,000 samples. It's way too much, so you're still stuck annotating, but you can optimize this annotation solution. You can increase the speed of it so that you, in the end, can train your model in a decent amount of time. And that's what we did. It was the project was called BioSegment, and there we did 3 things to solve this data problem. And you need to think about all these different things that can increase your annotation speed. You can have a better annotation by just giving the annotator a better GUI that can increase his workflow. You can interpolate the slices on the z-axis. So you annotate one image, you skip 5 slides, you annotate another one, and you interpolate the annotation between them. You can do sparse labeling with points and strokes instead of pixel perfect painting the whole cell pixel by pixel. And then you can also predict the refinement already coming out of your model instead of starting from scratch. We did more stuff on the training side, on the ML side. We can use unsupervised pre-training. You can train your model to reconstruct the unlabeled data, and then it has a better notion of what the data is like and can it helps in training the model. You can fine-tune an existing model, maybe trained on ImageNet, for example, on natural images. Then it has concepts that you can maybe transfer to electron microscopy images. You can use domain adaptive training. It's a certain loss function. And then you can also use active learning strategies to select better training data. So because we are in a low data environment, the choice of training data is really important. And instead of doing this randomly, we can spend some more time in picking out our training samples, and then these have better results when you train your data. So in this BioSegment project, we did 3 things. We compared 5 active learning strategies on 3 different data sets for this volume electron microscopy data. We created an annotation framework, BioSegment, with a certain software implementation to help annotators do this in one interface. And then we compared this workflow to 5 other annotation frameworks. And I can already tell you the 3 conclusions we got. So uncertainty-based sampling methods worked really well for our data. We used a certain Python framework, Fast API, together with PyTorch. It's a deep learning framework in our first version, and then we switched to MONAI Label and 3D Slicer. It's a type of active learning annotation interface used in medicine, and we created our own flavor for electron microscopy. And then we compare to other annotation frameworks. They're all actively developed. It's a really active field, but BioSegment worked well for us because it was tailored to our data, of course. Now, maybe I can again show you the active learning loop that the annotator goes through. So here at the bottom, you see the annotator with his annotator interface, and he starts a loop of 5 steps. So in the beginning, with 1 at the top, you start with a model. The backend applies this model to the unlabeled sample pool, and then you get the informativeness of all the samples. So then you just pick, it's just a ranking, you just pick the top sample, you send it to the annotator, your model segmented, the annotator refines it, you add it to the training data pool, and when you accumulate enough training data, you then start a fine-tuning job with your model. You get a fine-tuned model that is a slightly bit better. And then you can do this again. So now with this new model, you again do an active learning inference on your whole data set. And then you get a better ranking and you get better samples. So that's the loop that the annotator goes through. It's just a button for them, and it's a button get next sample. And in the backend, when we have enough samples, we change the ranking behind this button to get a sample with more utility for the annotator. Now, these 5 active learning strategies that you can do to get a ranking for your samples, there's like 4 different kinds. So in the top, you have entropy-based and least confidence selection. These just model the uncertainty of your prediction. You predict a certain class that this pixel is assigned to, and then you can get an uncertainty measure from this prediction. You sum it up for the whole sample, and then you get a ranking. You can also use a Bayesian approach. So there we have these dropout layers that you disable certain neurons in the neural network, and that gives you a kind of stochastic distribution, a bit of uncertainty in your model. It's very expensive to do for a segmentation model, but this gets you a certain kind of uncertainty that you don't get from the prediction. And then the third one is diversity from the embedding. So when the embedding is the summarization of the model, the highly informative data embedding. And with the segmentation model, with an autoencoder, you can use the middle layer of the autoencoder for this. So this is a layer with maybe 100 neurons, and you can do this lower dimension to do k-means sampling. And then you can get a bit of diversity. So images that look differently will be spread out in this embedding, and then you can pick different clusters from this embedding. That's k-means sampling. And then you can use a core set approach. It's a very niche approach that uses the embedding and also the labeled and unlabeled pools. Then we compared these strategies, and here you see a plot. We plotted uncertainty sampling versus random sampling in green over 4 active learning iterations. So on the x-axis, you see the 4 different iterations, and on the y-axis, you see the performance metric of the prediction. And you can see that orange, it starts out similar because we're only at a small amount of samples, but then it quickly grows to be better and outperform a random sampling. You can also do this for all the other uncertainty-based and the active learning strategies. And then you can see that orange outperforms blue in this graph. But these graphs are really difficult. It's really difficult to make a good graph for an active learning strategy. It was also a publication recently that you need a lot of automated hyperparameter optimization for each step. You need multiple models, which we didn't do here. You also need multiple data sets, which we did do. And you need a lot of regularization and statistical tests. But of course, this is for designing a new active learning strategy. Here we just checked to see if our findings were also correct if we use the labeled data sets to test this. And then for the second part, we implemented this BioSegment framework so that the annotator can do all these steps themselves. So we see an overview of all the data sets and the annotations on the left, and then a simple prototype viewer on the right. And this is a GIF of the prototype viewer. So here we use sparse labeling for 2 classes in the image, and then we inference on it in a 3D, you can slice it. But for the second iteration of this project, we used the MONAI Label and 3D Slicer. These are a bit more bespoke tools. And also because it's backed by NVIDIA and ITK, 2 big companies, it has a lot more development and we don't need to spend our own development time on it. And this is then the 3D segmentation tool in 3D Slicer. So we can see the result in 3D, and then you have all these different 3D tools to maybe tune this annotation even further. And you have a MONAI Label plugin on the left to get the next sample for you to annotate. And then finally, for the third step, we compare different annotation frameworks. So we compared these on 5 different topics: interactive fine-tuning, which they all did; active learning, which was not implemented in some, but you could add this in a plugin or in a later version. Support for large data sets was really important because we use really big imaging data sets. This 3D support that you saw on the previous slide, also pretty important if you work in 3D. And then remote resources, because we used GPUs, we really needed some computational power, and you couldn't do this just on the laptop of the annotator. And there we saw that BioSegment and MONAI Label had a lot of similar features. So then we created our own flavor of MONAI Label to have synergy for our own data. So these were our 3 conclusions. Uncertainty-based strategies worked well. We used and created an annotation framework, and then we compared it to the other frameworks. And then there's still an exciting future left for volume electron microscopy. There's this website, openorganelle.org, that really gives you a lot of ground truth label data for a lot of different classes of interest in the cell on a nanometer scale level. So we can use this to further pre-train models and then fine-tune them with our framework to get even more classes of your image segmented. You also have all these volume electron microscopy storage servers where you can store this data and pre-process it. And that's also pretty important to have more interoperability there, and they're actively working on it. And then to quickly process all this big data imaging, there's a lot of new frameworks out there to have better GPU support, chunked remote reading support, and better viewers to view this big data interactively at multiple resolutions. So a lot of cool stuff out there still. So maybe in the next update, we have more stuff on this. So for now, thank you for your attention.